is Professor Andy Ridgwell, and I'm very happy to see Andy here today. I've been lucky enough to work with him for years now, and Andy did his PhD at the University of East Anglia, UK, on glacial interglacial perturbations in the global carbon cycle in 2001, I think. And then he continued there as a researcher for a while before moving on to the University of California, Riverside. And then in 2004, he became an assistant professor at the University of British Columbia in Vancouver, Canada. And then in 2006, he went back to the UK, to the University of Bristol, where he eventually became a professor. And nowadays, he is back in California at the University of California, Riverside again, where he's now a full professor, and he still maintains his association with University of Bristol as well. And Andy manages the CGenie model and that community of model users. Yeah, as I said, I've been lucky enough to be part of that community for many years now, and it's a very active community, and Andy does a great and very vast job of keeping that going. So yeah, Andy, happy to see you, and I'm excited to hear what you have to talk about, because I don't know the topic yet.
Thank you, Malin. Let me get this screen sharing. I assume that everyone can hear me. You know, after 2 years of Zoom, I'd like to think that I can share screen and vaguely get it together. I mean, another thing to blame the pandemic and Zoom for, of course, is the fact that it's 7 a.m. here. So thanks Zoom for the very early morning time. This is not actually what I can see out my window. I live on the mountain opposite, which you can only vaguely see, but it was it's actually a wildfire camera for as we wait for the mountain to burn down. But it's pretty. And it has absolutely nothing to do with the ocean. So all I came up with for the talk is marine biogeochemical modeling, where next, which is a ridiculous title, because I'm not really the one to say where should we be going in terms of new model development. What those of you doing graduate study, what you might not realize, or you might have realized, it was something I realized early on in my PhD, the older professors don't know everything. That's a very important fact. You are the experts in what you're doing. And typically when you get older and more senior, you just do not have time to keep up with the literature. So really this title should be, you know, marine biogeochemical modeling, you decide. You know, from your work, you'll be the ones who have the, you know, the new ideas and hopefully new directions and make the new advances. Anyway, but here are some simple perspectives on developments in marine ecosystem modeling. So, you know, it's an understated, a complex system of many trophic interactions and species interactions and interactions between the environment and the ecosystem. So how do we go about trying to represent that system? I like to draw parallels with the sort of earlier days of modeling the ocean circulation. So I'm old enough that I remember when ocean box models were very important tools in understanding the global carbon cycle and the movement of nutrients, large-scale movement of nutrients around the ocean. If you put something in chat, one thing I've learned from Zoom is I can't read chat and do the share screen. So if anyone has a quick question, just ask it verbally. Anyway, so the ocean circulation is a complex system. You know, you have differentiating patterns of evaporation and precipitation to the ocean surface. You have big gradients in temperature, you have sort of deep mixing in some places, and a shallow mix layer in other places. What were the old strategies for trying to understand the ocean and how the ocean moves nutrients and carbon around? Like I said, in the early days, a lot of box models were made, which is fine. I mean, what you have to say, though, is that I know what the most important parts of the system are. So hypothetically here, thinking about a carbon cycle, you might have a box for the atmosphere, and then you might say, well, the ocean surface is fundamentally different from the deep ocean. The ocean surface is where the action is in terms of primary production and carbon uptake and nutrient uptake. And then there's a big box deeper down where all the carbon and nutrients ends up, but there's some sort of mixing term between the two. So you might conceptualize the ocean as just two boxes, the surface and the deep, because you said these are the most important parts. Or you might say, as a lot of people did in the sort of 80s when they were developing these ocean box models, mostly for carbon cycling, is that, well, actually, like the high latitudes are different from the low latitudes, and they're important. So maybe you want a third box, and now we can have some sort of circulation through the ocean as well as mixing between the surface and the deep. And you can keep adding boxes like this forever, and people did up to about 20 or 25 boxes, progressively saying, well, the North Atlantic is distinct from the equatorial Atlantic, which is distinct from the North Pacific, and et cetera, et cetera. So you can keep dividing up the system. You still have to be telling yourself, well, I know what the most important parts of the system are, and then you encapsulate that in the model structure. The problems are things like, what happens if you're wrong about how the ocean circulation works? It's a complex system. If you don't correctly identify the key elements and how they interact, then your model will be wrong. You might say, what happens in climate change? Future climate change, this is a fixed system of fixed fluxes and mixing. It can't account necessarily for, warming and transient stratification, which we're experiencing now. You might be interested, like I am, in what did the Cretaceous ocean circulation look like? It's like, well, this is the modern. You've taken everything you know about the modern ocean, and you put it in the model. You told the model this is how the system works, but then this is not then relevant for the Cretaceous. So what happened, of course, is people developed rather more complex models, but they didn't tell the models what to do. The key is they told the models what the underlying physics was. How does a fluid move on a rotating sphere under wind stress? What's the role of sea ice in brine rejection, in later on in melting and putting fresh water back on the surface? So you all know about these models. These are now, our primary tools. You can still build box models, but these now are models that are generic. They know the basic physics, and the circulation is an emergent property of the physics. And then you hope that the circulation is correct. You hope that you have an AMOC in the Atlantic. But that's not given, depending on the physics and the resolution and the boundary conditions. They're then equally useful for the future because you know about the physics, and if you heat the ocean at the surface, like we're doing now, they'll give you transient stratification. You can move the continents around, and you can run these models for the Cretaceous because they're just based on the same physics, and we're hoping that the physics of fluid movement on a sphere is the same in the Cretaceous as it is now. So these become very generic tools, and they're our main tools. So then what about ecosystems? And I can draw the same stupid analogy. Back in the day when people started first, worrying about how to represent the uptake of nutrients and carbon, et cetera, at the surface, a very influential models developed at Southampton were single nutrient, single phytoplankton, single zooplankton, NPZ, and you can add the trisis, which is the D. So you could look at this as two boxes. There's a box for the phytoplankton, the primary producers, and the box for the zooplankton. You can keep adding these boxes. You can say, well, actually, diatoms are distinct from other phytoplankton, so we can have two phytoplankton boxes, and we can have one zooplankton box. And you'll sort of parameterize what these phytoplankton do, typically after modern experiments and observations. And you can keep adding boxes. And some of the CIMS models have, I think, up to 10 different functional types of phytoplankton. So 10 phytoplankton boxes and a couple of different zooplankton boxes. But you can make the same criticisms with this approach as the ocean circulation box models. You can say, well, you're encapsulating in a model what you think the answer is. You have to know how the modern marine ecosystem works before you can devise these boxes and make the links between the boxes. And so what happens if you're wrong? What happens if you do not actually fully understand the modern marine ecosystem and its dynamics? You can also say, well, what about climate change? Will these things respond? And I'll come to that in a few minutes. Does such a sort of box model appropriately respond to climate change? And then you could say, maybe, well, what about the Cretaceous? There were phytoplankton in the Cretaceous, so that's good. You go back to the Jurassic or the Triassic, though, so this is about 200 million years ago, and there aren't any diatoms. So if you have a model where you say today diatoms are key, and then you go back to the Jurassic and you want to understand the carbon cycle in the ocean, then well, you can't have diatoms. And it's like, well, if you've taken away one of the two key boxes, does the box model still work for the Triassic or the Jurassic? The answer is probably not. So then, can we create marine ecosystem models that have a sort of parallel to our main ocean model tools, which are the ocean general circulation models? And the answer is yes, we can start to build much more complex and flexible models. This is just something I pulled off the website for the Darwin Project. I would say that this is led by Mick Follows at MIT. I would say that a talk I heard at a conference from him about 15 years ago was the most influential talk I've ever heard in my life. Just how you can generate random ecosystems of many, many, many different possible phytoplankton and let the environment select what is the, spatially, what is the competitive and what is the fittest combination of phytoplankton that should be in that ecosystem. So if you're not aware of this model, I encourage you to go Google Darwin Project MIT and read some of the papers behind it. And this sort of model relies on, which is difficult, identifying the fundamental rules behind plankton. So for the ocean circulation, it's sort of easy. The fundamental rules of physics, the physics of fluids, that's simple. The rules of phytoplankton, there aren't really rules of phytoplankton. You can't model the folding of proteins and the synthesis of amino acids and the fundamental physics, if you like. You can't model these from first principles for every cell, but you can draw some, you can sort of create some empirical rules, for instance, to do with size. So the one that I guess as someone who isn't really a marine ecologist, the one I sort of understand the best is to do with size and nutrient acquisition. So there is a fundamental rule, if you like, between the size of your cell and how easy it is to get nutrients. So if you're a small cell, you have relatively large surface area compared with the volume. So your metabolic demand is relatively low, and you have relatively lots of surface area for instance, some nutrient transporters on the cell surface, and you can acquire nutrients at pretty low concentrations. If you're a large cell, you have a large demand for carbon and photosynthesis and nutrients, but proportionally your surface area is small compared with your larger volume. So you have a problem stuffing enough nutrient transporters onto your cell surface. So large cells tend to have problems acquiring nutrients at low concentrations. So one sort of rule is a sort of trade-off between cell volume and nutrient affinity. And you can sort of create some other sort of rules, pseudo rules, empirical, observational based sort of rule like relationships like this, and build your model. using these rules rather than defining very specific plankton types. Well, that's fine to an extent. I think the Darwin model, they typically have no more than 70-something possible phytoplankton, of which most turn out to be uncompetitive. But the ocean has rather more than 80 different species in. So how do we deal with the complexity in terms of sheer species numbers of phytoplankton and zooplankton in the ocean? So one trick that the CMIP models use is noting that, so if you look in this bottom right-hand plot, it's a cartoon of some different temperature growth rate response curves. And obviously I've drawn the cartoon that you can see that there is a sort of a smooth, you could draw a line through the maximum of all those dotted curves as a function of temperature. And that's exactly what people did way back. So in a famous paper by Epley, they noted that from culture experiments of all sorts of different plankton, you can do exactly this. You can put this line through the maximum, and then you can say, well, rather than represent all these different individual species, all these different temperature optima and crash your model because there are so many phytoplankton and so many traces you have to move around the ocean, why not just say, well, we can approximate this with one plankton that has this temperature growth response that follows the epley curve, or a curve very similar to the epley curve. This is like having a superbox. You're saying, of the 10 or whatever it is, 1,000 different species of plankton in the ocean, we can just define a couple of sort of super plankton to put into the model. So what are the consequences of this? So a series of little cartoons. Here I'm thinking of a particular, say, high latitude environment. So range of temperature given by this little gray box. And the thick line on the epley curve just represents the range at which plankton would survive in that environment. So if you have warming like we have now, or if you have warming like we had in the geological past, you could say, well, we just move up this curve, so whatever plankton there are just growing slightly faster. And then if after we burnt all the fossil fuels we can, and the Earth gradually recovers, and the system cools back down again, you just follow smoothly this epley curve, this function of growth rate versus temperature back down again to this sort of last bottom panel where, hypothetically we reestablish the initial climate and the ecosystem is exactly as before. So here we have a system where, in response to a big perturbation like we're doing now, you smoothly increase all the growth rates, and then as you recover the system and the Earth cools back down, you smoothly go back to slower growth rates. It's completely reversible. No disaster has occurred here. So I thought that we were worried about climate change because of the risk of extinctions, and extinctions we're seeing now. And so here is a model system that cannot do extinctions because however bad the global warming is in the future, you can smoothly reverse it back down again. What if we resolve like a range of individual plankton? So this is more like the little bit more like the Darwin model. You will lose the cold adapted ones as you warm the system. But if you say, well, these things are always there, so if in the model you always have some plankton that are cold tolerant, then as you cool, you start to recover these same colored curves in the same places again. And you reestablish when you reestablish the initial climate state, you reestablish your ecosystem. And this is known as everything is everywhere. So i.e. every possible temperature optima plankton is somewhere. And you select for it, and if it warms up, you select for the warmer ones, and if the system cools down, you select for the colder ones, because everything is everywhere, or the ocean is fully connected everywhere at the surface. So what about, I mean, I think this looks like more the real world, and this is where I'm interested in the system and how to address it and how this might help us understand past extinction events. So we start with the same system. We're thinking about things living, say, in the Southern Ocean. You have a warming, so they're going to go extinct. So if you have a whole bunch of cold tolerant species in the Southern Ocean and it warms right up, they go extinct. When you cool back down again, they're not there because you've had an extinction. Eventually, when you've reestablished a cold Southern Ocean, so in the very bottom right-hand plot, you will repopulate the niches, if you like. You will re-evolve plankton that are cold tolerant, but they don't necessarily have to be the same plankton. They don't have to have the same temperature optima. They don't have to have the same properties. So your reestablished ecosystem is likely to be different, which may have interesting implications then for carbon cycling and nutrient cycling and everything else. And this is, on the right hand, this is the sort of system I'm interested in, and can we model this system? So I've got just, I don't have many minutes left. I've got a couple of little animations. I'm going to go through this fairly quickly, and it concerns what's known as trait space, little cartoon on the bottom left, and how fit you are in that trait space. So my sort of stupid little example I like to think of is so it's not a marine example. So there's a gap in the forest. Let's say there's a space, there's a glade amongst the trees. So there's no big shade trees. And if you're a terrestrial plant, then you have the ability to grow in that space and do well. And let's say that soil is highly nitrogen limited. So you need to be good at nitrogen fixing to be able to take over that space. And so as you develop or as you evolve a better symbiotic relationship in your roots with nitrogen fixers, you move along one of the trait axes, and then maybe you move up one of these hills. So you become fitter and fitter compared with everyone else, because no one else can do nitrogen fixation as good as you, and the soil is nitrogen poor. So it's not, the key to this trait space is you're not moving like physically, spatially, you're moving in terms of your abilities to be successful in the ecosystem. So here's a really weird little example I found, little animation I found from the internet. So I guess that they did this in MATLAB by the looks of it. And this is like two different trait axes. So here, two different abilities. One might be ability to nitrogen fix on one axis, the other might be the ability to grow tall and shade out other plants. And these peaks just represent what combination of traits, so what combination of ability to nitrogen fix and ability to grow tall gives you the most competitive advantages compared with all the other plants around. And so the point is there's some mutation and selection, and gradually your population, or your, I guess here it's there's a variety of different trait combinations, different sort of genetic, there's some genetic variability amongst these different disturbing little bugs, and they sort of evolve up this this peak of fitness, and all the other ones go extinct basically as they're outcompeted by the increasingly fitter ones. I'm interested in this system because the real world is not like this at all. The real world looks even weirder in this MATLAB animation, because if you think of your gap in the forest, you're good at nitrogen fixing, you grow, you outcompete everyone else, you keep fixing nitrogen, you're going to start adding nitrogen to the soil. And as you start adding nitrogen to the soil, you change the fitness landscape. So if you look at these bugs, they crawl up one hill and the hill goes down, because as they conquer that hill with high rates of nitrogen fixation, and you add nitrogen to the soil, it's now no longer competitively advantageous to put all your resources into nitrogen fixation because you've added nitrogen into the soil. So then you might want to evolve, or some other populations move in that don't bother doing nitrogen fixation because you added the nitrogen and they outcompete you. So the point is, there is this really sort of interesting feedback and relationship between population evolution and the evolution of the environment. If you want to coin a sort of a cheesy, you know, proposal funders, funding agency phrase, the co-evolution of life in the environment sort of looks like this in weird MATLAB animation space. And I'm interested in, you know, can you build a model to simulate some sort of evolution between life, so genetic evolution of a population and the change in environment that results from an evolving population. So the final slides, this is work of Ben Ward.
Andy, there's no rush because we started a bit early, so you can.
Are you? I'm almost done. I mean, I've got, all right. Well, I'll dwell on this because it's weird for 5 minutes if you like, and then I'm done. Yeah,
sounds good.
So this is work of, I mean, the only slide I skipped over was just thinking of, because my interest is more geological timescales, and there have been fundamental evolutionary transitions during Earth history. The advent of oxygenated photosynthesis was maybe the most fundamental evolutionary event that ever occurred in the history of the Earth. The evolution of animals was another fundamental transition. You know, the ability to now eat lots of smaller than you cells was another fundamental transition. So I'm interested in both how some of these evolutionary transitions affected the environment, and also how a changing environment would affect that trait, that fitness landscape. So as oxygen accumulated in the atmosphere, or as ecosystems started to be grazed down by the newly appeared animals, you know, what sort of selective pressure did that put on further animal evolution, for instance? So I'm interested in this sort of the feedback between, like I said, the evolution, the biological evolution, and environmental evolution. Anyway, so this is like version 0.001 of a model that can do the co-evolution of life in the environment. So this, like I said, it's work of Ben Ward. It's actually written in MATLAB. It has an ocean transport model, but it's been turned into, if you've come across the matrix transport approach to modeling how the ocean moves around, for instance, carbon and nutrients and things. So this is the trait space. On the x-axis, we have, I didn't talk about mixotrophy. So we have two end-member strategies as a plankton. You can be on the very left hand, and you can be a pure photoautotroph. So you only get by photosynthesizing. Or on the right-hand end of the x-axis, you can be a pure heterotroph. So you get by only by eating other people, and you can't do anything else. The ocean's a real mix of things. I mean, on land, plants that can also digest insects, a few. There's the sundew, there's the Venus flytrap, and I guess the pitcher plant, I think, are the only ones I can think of. So it's very rare to be a mixotroph on land, but it's much more common in the ocean. For instance, I don't know whether it's all dinoflagellates, but dinoflagellates are a key modern mixotroph. So here are like two end-member possible strategies with everything in between. On the y-axis is different possible sizes your cell could be. Why is size important? Well, if you're larger, you could eat larger prey. If you're smaller and a phytoplankton, so if you're up here in the heterotrophic end and large, you could eat all sorts of things. If you're down here, for instance, and you're a pure phytic autotroph and you're very small, you can acquire nutrients at very, very low concentrations. Up here is a large phytic autotroph. You're photosynthesizing and you need a lot of nutrients around. If you're down in the right-hand corner, you're a small heterotroph, and you can eat things, but they've got to be very small. So you may be limited in this bottom right-hand corner. And the idea of this model is to, at least this example experiment, is to put into the ocean one single functional type, if you like, one single species, which is going to be a tiny, I can't remember what the cell size is, but the smallest possible size in the model of pure phytoautotroph. So the model is starting with nutrients everywhere, and it's seeded with a single phytoplankton, a single very small functional type of phytoplankton. And then the model is allowed to evolve, and there is this sort of pseudo evolution that's allowed. If you're a successfully growing population of a certain species, so for instance, a small phytoautotroph, a very small amount of biomass is put into a slightly more mixotrophic form, and a very small amount of biomass is put into a slightly larger phytoautotrophic form. So you're diffusing your traits into some biomass adjacent. So this is a little bit like mutation. So you can imagine a mutation that it gives you the ability to very slightly consume other phytoplankton, and a mutation that gives you, going up the y-axis, a mutation that allows you to develop a very slightly larger cell size. So it's a little bit like mutation, but not quite. It's a simple approach, but one that is gives some rich outcomes. You can't see these things on the screen. There's little postage stamps of the global distribution of biomass. So we're not going to worry about this, but these are actually almost 1,000 little pictures of what the distribution of each of the functional types is in the model. But like I said, it starts only with one that exists, and these things can evolve and they can go extinct. So it's not that we seed the model with all 1,000 possible phytoplankton. We seed it only with one tiny one, and we let the system evolve, mutation and selection. This is the visualization of what the animation I'll show you in a minute to finish off looks like. On this axis here, the trophic strategy is the autotrophy to heterotrophy, so the degree of mixotrophy. On the y-axis again is the size of the plankton, and then along here, I assume you can see my mouse, is time. This is in years of model time. It's obviously rather fast for evolution, but like I said, it's an exploratory sort of test, proof of concept sort of model and simulation. These branches are just the biomass of each. If you want to call it a species, fine, each species. So it starts at time 0 with only tiny phytoautotrophs. Immediately the system starts to evolve to larger phytoautotrophs. And then it starts to move, things start to evolve to a more heterotrophic strategy. So for instance, going into this purple, the initial history of the phytoplankton is as a pure phytoplankton. It grows to be a large phytoplankton and then gradually evolves to be a large zooplankton. I like this because we have extinctions. So here is a branch of large mixotrophs that go extinct because they've been outcompeted. Ultimately, the better strategy was to be a really large heterotroph. You have no baggage of trying to photosynthesize and take up nutrients when the nutrient concentrations are low everywhere. So here is extinction. Here is now I can't remember the term. I'm not going to pronounce the term. It's embarrassing. Convergent evolution. So here there's a branch, the white separates from the green, but gradually they converge back to, if you want to call it the same species again. So this is convergent evolution. So actually, you can play out some what look like real quotes unquotes evolutionary games in this model. And the animation looks like this. I'll let it play through a couple of times, and that's me done. So when it starts again, the first thing you'll see is this spread of biomass on the y-axis upwards. And this is because the model starts with nutrients everywhere. So there's a rapid pressure, there's a pressure to rapidly develop to be large phytoplankton because there's plenty of nutrients. The nutrients start to run out. Maybe I have to press play again. The nutrients start to, oh, what an amateur. The nutrients start to run out. So then it becomes a good strategy to start eating your friends. So this movement to the right is the evolution of more heterotrophic strategies, because there are lots of people hanging around with lots of nutrients in their cells, and why not eat your friends? And then it develops these larger cell sizes of zooplankton. I'll do it once more, if I can get it to once more without going to the last slide. And there's like a large mixotroph that develops in this sort of top middle section that does all right for a while, but eventually goes extinct because it's outcompeted by the large zooplankton on the right-hand end. So there's biomass here. It does all right for a while, but ultimately the best strategy for consuming large prey is just to concentrate on the heterotrophy and not try and take up nutrients from solution. So that's it. I mean, this is not an answer to anything. I'm impressed that we can even get something that looks like evolution going on in a model, and an interaction between plankton evolution and the environment, which in this case is the nutrient cycle of just phosphate in an ocean circulation model. It's not necessarily the direction we have to go in developing new marine ecosystem models. Like I said, the older of us, you know, we're running out of ideas because we don't tend to keep up with the literature. So it's in your graduate study years and maybe your initial postdoc years that I'd say that you are at the peak of your expertise and the peak of your ability to come up with new ideas and new directions in modeling of marine systems. So I would be confident. Don't think that older professors know everything, because actually we don't. So I'll leave it with that message.
Thanks a lot, Andy.